Hello, here is a pre-lecture on diffusion and osmosis to be ready for Lab 5. We're going to be talking about solvent, solute, and solution, osmosis and diffusion obviously, and some factors that will influence the rate of diffusion, selectively permeable membranes, the tenacity of a solution, isohyperhypotonic, cell walls, and then hopefully some applications of these things. You're also going to learn how to measure liquids in metric units, milliliters in particular. First up, solvent versus solute. The solvent is the dissolving substance. The solute is the substance dissolved. So in this beaker here, looks like there's a liquid. That would be the solvent. It is often water. The solute is the substance dissolved. In it, these little yellow dots, together that makes a solution. So if we're talking about lemonade, right? If you're making lemonade with a mix, you put some water in the glass, that's your solvent, and then you take a spoonful of lemonade powder that involves some sugar and other things, and that would be the solute that's dissolved. Together, the solution is lemonade. Diffusion is the movement of molecules across the membrane in particular from an area of high concentration to low concentration. Molecules are always moving, even if we can't see them moving. Even in a solid, they're moving. And particularly in a liquid or a gas, they are moving around a lot. And they go from an area of where there are lots of them to where there are fewer of them without, just because of random molecular movement. Okay, so if we put a drop of water, of food coloring into water, it's going to start off as a very tight, concentrated drop of liquid, and then slowly it disperses throughout the entire container. Eventually this whole thing will just be blue, right? And that is just because the molecules are moving them around. They wiggle, they do this little molecule dance, okay? And they're moving from that area of high concentration in the initial dye to spreading out throughout the whole container, where they're then in a low concentration relative to the other molecules around them. Because scientists like to have different words for different things, and this really is a different thing, so it should have its own word. Osmosis is the diffusion of water. So water molecules moving from an area of high concentration to an area of lower concentration of water molecules. And those are two very important words that you should know. Now, this gets really exciting when we're talking about this membrane in between two areas of a solution. Now, as you know, plasma membranes are cell membrane around a cell, so is usually what we're talking about, and not in a beaker. But it can have interesting impacts on the shape and size of cells based on where the membrane is. So let's do a real quick review of a plasma or cell membrane. Right, we have the phospholipid bilayer here with the hydrophilic heads and hydrophobic tails in between. And then these proteins that extend through, they have the hydrophilic areas and hydrophobic areas, so they are suspended within the mosaic. And the role of the membrane is to keep certain things in and out and regulate transport across the membrane. Right? There are some things that can move directly through the phospholipid bilayer if they aren't polar. Other things need to move through a pore in a protein. I think my next slide is just that. So here's a pore protein. So there are a polar molecule could pass through that channel from this area of higher concentration to the area of lower concentration. Just briefly, there's also a carrier protein, which is changing shape to move a molecule through into or out of the cell. And this can also work against the concentration gradient, but we'll talk about that in a minute. Depending on how much energy is used is how we determine passive or active transport. Passive transport does not need any energy added. So carbon dioxide and oxygen can diffuse across a cell membrane because they're not polar. They just go right through the phospholipid bilayer. Water is polar, so it needs to use a protein channel, but it still doesn't take any energy to do that. When we're pumping against a concentration gradient, that's when, and we therefore need energy, moving molecules against the direction they want to go in. That's active transport, and we needs energy. We saw another example of active transport was the phagocytosis. The amoeba was engulfing the paramecium. That obviously takes a lot of energy to extend pseudopods and enclose and engulf and reform membranes, and then, but obviously there's a benefit to that. That's how the amoeba eats. As it captures its prey, but that's a different kind of active transport. All right, I want to talk about osmosis and water and cells and solutes. So, in terms of the tonicity of a solution, there are, you'll see in these images, cells can either be hypotonic in relation to their solution, they can be in an isotonic solution, they can be in a hypertonic solution. And this has to do with the relative concentration of solutes inside and outside this cell. Let's start with hypertonic. Hyper means more than, like hyperactive or hypervigilant, right? Hyperactive kid is bouncing all over the place, has more energy than most people. In a hypertonic solution, there's more solute on the outside of the cell than inside. And you can see this in this image because there's more green dots out here than there are inside. Okay. Over here, in the hypotonic solution, hypo means lower, right? Like hypoglycemic means you don't have enough sugar in your blood. Hypothermic, you're too cold. So there's fewer solutes outside than inside. So, isotonic, iso means the same, so there's the same amount of solutes inside and outside. Now, why do we care? The caring bit comes from what happens to water and then cell structure in all of these things. Okay. In the hypertonic solution, there's more solutes outside, but that means that there's a higher concentration of water on the inside of the cell. And water, as we know, can pass through a membrane. It's permeable to water membrane. So if water is moving from the area of high concentration to low concentration, that means that the cell is going to lose water and get smaller. Over here, there's a higher concentration of water on the outside than on the inside in the hypotonic solution. And that means that water is going to go into the cell rather than out of it, because there's a higher concentration outside, so the water is going to move in. In an isotonic solution, there is water moving in both directions, but there'll be no net gain or loss because the same amount of water will be moving across the membrane in either direction. Right? So now if we look at some images of this, the animal cells here on the top, in a hypotonic solution, if it's the wrong tonicity, enough water will go in that the animal cell with its lonesome cell membrane will actually explode or lyse. Right? In a hypertonic solution, that animal cell will shrivel and shrink. Okay? Isotonic, equal in and out here, so it stays the same side. So when we are treating a dehydrated patient, animal or human, right? If we give them pure distilled water, that hypotonic solution would make the red blood cells explode. Right? We give saline, which is a little bit salty, not like not drinking salt water salty. That's a bad idea, right? But saline solution, of whatever percentage is needed, is isotonic to the blood. And so it returns some water to the dehydrated cells without lysing them or causing them to shrivel. Now, plant cells are a little bit different, and we're going to check this out for reals in lab with the Elodea plant. But as you remember, plant cells have a cell wall, and that cell wall keeps them from exploding. So in a hypotonic solution, the central vacuole, which isn't shown as well in this picture, but that's okay, is full of water, because all the water is rushing in, and that pushes against the cell wall and helps keep the plant upright. We call this turgid, but it's a normal plant cell prefers to be in a hypotonic solution. There are, of course, are exceptions of like saltwater plants, but for the most part, plants prefer as pure water as you can get. Right? In an isotonic solution, where some water is leaving the cell, the plant can become flaccid, which it's not fully wilted or dehydrated, but it's not, because the vacuole isn't as full as it could possibly be, it's not pushing as hard against the cell walls, so the cell is turned flaccid. In a hypertonic solution, the cell membrane actually, because so much water is leaving, the cell membrane pulls away from the edge of the cell wall, and then you can see, and I will finally prove to you that there are cell membranes in plant cells, even though you couldn't see them before when we look at that in lab 5. So these are, schematics, but important to think about in terms of how cells react. Okay. The other thing I want to go through with you briefly about is directly related to the lab, learning to measure in volume, and then we're going to be looking at the conductivity of salt water. Okay. So when we're measuring in metric, we're using milliliters, and the important thing to know about is using this piece of equipment, which I'll show you, is a graduated cylinder. We need to look under the curve of the meniscus to read it. Right, and I'll do a full demo of that, but I wanted to show you this picture right now. And then I also want to talk to you about conductivity of water. In plain water, with plain water, this is an experimental setup so that you'll believe me that water can conduct electricity, if I can get it to play. All right, so if we connect the ends of these wires, the lights up. See that light bulb? So it's got voltage attached to it, and a 100-watt bulb, and when the wire's connected, it turned on. So now we need to, if the electricity can pass through the water, the light bulb will light up. Okay, so this is just plain distilled water first, and as you can see, the light bulb is not. But if we add salt to this water, we put in sodium chloride, which is an ion, and we get enough in there, so keep watching that bulb over there on the left. You see how it's starting to turn on? They need some more. More salt, more salt. All right, and it's starting to light up. So because there are ions in the water now, the electricity is able to travel through it and light up the light bulb. All right, so when we put that sodium chloride into the water, it splits apart into positive and negative ions with the water, because water is a polar molecule. It's a universal solvent. So we're going to be putting the salt solution and starch solution into a tube. So here's what the salt ions look like. Here's the starch molecule. And we're going to see which of these molecules can diffuse out of a cell, and if it is moving through, if it is diffusing through, then we will have increased conductivity in the water outside. See you in lab.